am excited to be here today to the, it's like 30 of you left to talk about THCV and some plants that we're developing that are high THCV. Let's see. There we go. So Phylos is a crop science company. We are developing new cultivars and also seed-grown plants that are uniform and can be planted out in a field or your greenhouse. Not pheno-hemp seeds, but ones that will give you consistent results. And we've built this program off of evaluations of tens of thousands of cultivars. We have great relationships with some legacy breeders and have in-licensed some of their varieties and pay royalties on those varieties as we use them in breeding. I think that's important to mention in this industry. We also have a pretty big focus on minor cannabinoids and trying to bring those into the majors so that we can see high levels of CBDV, THCV, and others. And we have a pretty serious focus on customer support for new growers that have been growing all different kinds of plants at their farms before and are ready to move into cannabis. And so we're trying to bring them into the world of growing cannabis. So today I'm going to talk about THCV. I think a lot of you are probably very familiar with this cannabinoid, but I want to talk just a little bit about what it looks like. So THC is on the left, very familiar. There are only two carbon differences between THC and THCV, so it's just got a shorter side chain than THC. Makes a pretty big difference in how it interacts in the body and what kinds of receptors it binds to and how it activates those receptors. But they are very similar looking, and you'll see in the pathway for creating these cannabinoids, they go through exactly the same pathway, same enzymes. So this slide actually, I would say THCV is a really promising cannabinoid. I don't know if it's the most promising cannabinoid. I mean, CBDV is amazing. You saw talk about CBG today for topicals and skin irritations. I think that there are so many cannabinoids, like ones that Dedi Maria is talking about, that no one even knows about that have potential, even at super low doses, for being important in medical applications. So, correct that slide and say that it's a promising cannabinoid. So there have been a few clinical trials. There's been a fair amount of preclinical work in laboratory and rodent models to look at the potential of THCV. So this is a listing of papers that, or studies, clinical trials that have shown positive responses for different diseases or afflictions. There are some that have negative results, and I think that the conclusion that I've come to is that dosing is really important, so we need more studies, more doses, really looking at what it takes to get an effect and where this cannabinoid is going to be useful. So you can see that there's evidence for anticonvulsant activity, could be useful for epilepsy, from a Hill et al. Paper in 2010. A lot of people talk about THCV as being skinny weed, and I think the jury's still out a little bit, but there definitely seems to be better control for blood sugar, some other markers of diabetes, insulin sensitivity, glucose tolerance, and then there is one study that shows that there are appetite suppressant effects, although there are other studies that show that there is not an appetite suppressant effect. There is a study that shows that there may be improved social and cognitive behaviors in a schizophrenia model, and then also improvements in neuropathic pain. And finally, something that I think is really incredible is that it's a potential for treatment of PD, but also for reducing disease progression. So not just symptoms, but possibly important for slowing that disease down. So, like I said, these are a lot of preclinical trials, very few clinical trials. Some of the clinical trials have used very small doses, and I think that, like I said before, dosing is very important and we need to look at more doses. They're doing a clinical trial right now just to look at safety and tolerance for up to 200 milligram doses. So that'll set the stage for doing clinical trials with more levels, more arms. And then oftentimes in the rodent models or in the cell-based systems, they're using really high doses. Actually, I list the Cascio paper, and that's 2 milligrams per kilogram, which is not super high for somebody like me. That's a 60 milligram dose. Yeah, it's kind of high. But some of them are like a 25 milligram per kilogram dose, and that's like 1,500 milligrams of THCV. I'm not convinced that that's going to be realistic in a human model, in a human clinical trial. So I think that we need to do more studies, more clinical trials, look at different doses. I think that there is a recreational potential also for THCV, and we're doing a little observational study right now to look at responses to being active or sedentary, to being interested in food and what your appetite is like, and then looking kind of at just a sense of joy and how you're enjoying a particular product with an even ratio 1-to-1 of THC and THCV. So we're doing that. It's small, but I think that we also need a lot more observational studies to look at the effects of THCV as a recreationally important cannabinoid. So the problem with THCV, though, is that it's typically found in really low percentages. There are reports of some cultivars having up to about 7% THCV. When we went out and looked at what was out there, we were seeing, obviously you see many varieties that have 0, but for the ones that had them in some level, it was about 0.5% to 4%. So still pretty small amounts, and so if you want to make a full spectrum extract and put it in some kind of an edible or something like that, you're still looking at having to produce a lot of biomass to get to that level of extract that would be viable as a product. So we would like to bring this cannabinoid into the majors. We think that it is important for medical and recreational applications. But what do you need to do that? You really need the trait to be heritable. You need to have natural variation for the amount of THCV. We do marker-assisted breeding, but that's all on natural variation. Need it to not be phytotoxic when it goes to higher levels. And we need to understand that chemical pathway that goes from the precursors all the way down to the cannabinoid so that we know where to look for genes that are important for making this cannabinoid. So I'll start with the pathway. So I'm starting at the top and so you have these two precursor molecules, butanoic acid and hexanoic acid, and those eventually get down all the way to the bottom, and you get your either pentyl cannabinoids, the ones that we talk about a lot, THC, CBD, CBC, or you get to the varins. And so they start either with the hexanoic acid for the pentyls or the butanoic acid to get down to the varins. Next step, so they're using the same enzymes to get there. So they have these different starter molecules, and then they're using acyl activating enzyme with the different starter molecules to get to the next step. So you can see already they have this difference in this carbon tail. So right at the beginning, you've already got the difference that you're going to see in the end product. And then you add, another molecule to malonyl-CoAs, and you add 2 more enzymes, and they're these same enzymes. And so then you get to divarenolic acid or olivitolic acid, depending on which starter molecule you had. Final step is GPP on a parental transferase, and taking those down to get to, I'd say, the mothers of all cannabinoids. So we have CBGV and CBG. And then that final step is you're using these same 3 enzymes, these same cannabinoid enzymes to get to the final products, either the 3 varins or the 3 cannabinoids. And please, I know that I'm simplifying this a lot. There are probably, 140, 160 different cannabinoids that we know of now. I'm only talking about a few of them. But what I'm just trying to illustrate here is that, we start with these 2 different molecules and then use all the same components to get down to the bottom. So what happens to get to those first bits that seem like they're the important determinants of the kinds of cannabinoids that you're going to make? We have to go all the way back up to the fatty acid elongation process. And I don't want to go through this whole thing, but the important thing is that it's starting at these really base molecules. And for butanoic acid to get to the varins, it only goes through the cycle 1 time. So it only adds the 2 carbons on the end. But to get to hexanoic acid, to get to your standard cannabinoids, it goes through the cycle 1 more time, just to add 2 more. So every time it goes through the cycle, it adds 2 more carbons to the end. So this part of the pathway, which sits on top of what I just showed you, is what's really important for making those precursor molecules. So, bottom right there, understand the chemical pathway. I think we have a pretty good handle on that. It's some really low-level stuff, right? This is important stuff for the cells. And then for these other things that are really important for us to bring this cannabinoid into the majors, we see natural variation for THCV. We've seen it all the way from a range from 0 to about 6%. In our hands, we only had 0 to 4% to start with. We see it as a heritable trait, so when we look at a family, it segregates in the family, so you see some plants with none, some with a little bit, and some with more. And then for phytotoxicity, we're making some assumptions here, but we know that THC can go to 35 or more % of the plant by dry weight and is not phytotoxic, so we think that for THCV, we'll have no problem getting that to a higher level. So we use marker-assisted breeding. We find genetic markers that are associated with the trait, and then we can use that to grow a lot more plants than we could by standard methods. So if we have these markers that are associated with the trait, we can grow tiny little seedlings in 0.2 square feet per seedling plug instead of having 2 square feet per mature plant. And we can get 50 times more plants processed in that way than if we had to grow them all to maturity. So probably telling you things you already know, but it allows us to go faster because we can grow more plants. So after we have tested them, then we're able to select for those plants that are predicted to have high levels of THCV, grow them to maturity, and test them. So the first part of this is discovery of the genetic markers. And this is an example just with photosensitive and day-neutral plants that I wanted to go through just really briefly to kind of describe what we're doing. But we take a plant that, say, is photosensitive, one that's day-neutral. We cross those. The F1 plant there is heterozygous for being photosensitive and day-neutral. And then you self that plant. All the chromosomes get mixed up. Recombination happens, and you can look for plants that are day neutral and look at what genetic markers are associated with that trait. And so you can see here we've got these 2 little day neutral plants, the little p's, and they both share a marker that on both of their chromosomes that allows them to be day neutral. All right, so we're doing this with THCV, crossing a no THCV plant with a plant that makes some THCV, and then selfing the F1, and same kind of thing. Letting genetics do its work to help us identify this association between the phenotype and the genotype. All right, so our first shot at this, I feel like we got kind of lucky. So this was a population of plants that did not make very much THCV at all. Initially, that wasn't even the point of doing this, of making this family. But we made this family, and it's segregated for having no THCV versus having some. And we used QTL mapping on this family. First made a linkage map with all the individuals, and then we had cannabinoid data on 142 of those, so we were able to make a QTL map. We found one really strong peak. I was shocked. Didn't expect for this to happen. And so we started taking a look at some additional germplasm that we had. You can see that for the QTL map, you get, with 142 individuals, you have this giant swath of a chromosome. Hard to pin down exactly what's going on. So what we needed to do was to go into additional germplasm and look for, and use the plant's recombination to mix up all these, all the genetic information so that we could really see what's associated with the trait. And so we had 302 accessions from 67 different seed lots, did nested association mapping. That's like a combination kind of between QTL mapping and genome-wide association studies. It's making use of family information, but it's also making use of the fact that you have lots of recombination between the different families. And of those, they still had pretty low levels of THCV, but we were able to confirm that spot on chromosome 4 and could start to narrow down what gene was likely responsible. And it turns out it's a gene called KR or FABG, and it is part of this fatty acid elongation chain pathway. So we got really fortunate with our populations, even low levels, we were able to do this, low levels of THCV, and then we were able to identify a gene that is in the pathway very likely to be the gene that is responsible for having some production of THCV. I should note that an Australian group also reported on this at relatively the same time, and voila, they found KR as well. So 2 independent groups finding the same gene. All right. So what do we know? We know that in order to produce THCV or CBDV or CBGV for that matter, all the same precursor, you need to have this particular allele of the KR gene, and that you can have either 1 copy of it or 2 copies. And then, we had very low levels of varins, but we know that they get much higher. So what explains much higher levels besides just making more cannabinoids? Well, it must be other genes. So we knew that we needed to do more work. And so we looked at additional small families, same kind of process. We also had another F2 population and evaluated those in the same way. We also did have some African landrace seed lots that showed some segregation for varins, which is where we think that the initial varins were located, was in southern part of Africa. So we did that, and what we required is that all of the plants had the KR allele that would allow for production of varin. And so we first restricted to that. So we knew that every plant should produce some THCV or CBDV, but what we wanted to get at were additional genes that would elevate that cannabinoid. And so we found genetic markers on 5 additional locations, chromosome 1, 4, 6, and 2 markers on 7. 7 is where we typically find the cannabinoid synthase genes, although different location on the genes. And we had, in some of our crosses, 2 accessions that were positive for the markers that we found, and they contain between 10.5% and about 11.5% varins. So that is good news. So what are we doing? We are continuing this cultivar development of high THCV and CBDV plants. We have done additional validation and additional crosses, and in our validation we found that one of the plants had all the markers, it looked so positive, so promising, and then it only made 4.5% THCV. So it's clear, and then sort of another thing is that we had a plant that made 14% THCV. And it only had 4 of the markers. So very clearly there's something else going on. There are other genes that are important. There's more work to do. We're continuing along this path and hope to nail this down and be able to share more updates as we find them. So the next steps, I kind of just summarized that. We expect 6 genes at least to be involved in this pathway. We think that they're involved in that sort of upper part of the pathway where you're making the precursors and it makes different ratios of the hexanoic to butanoic acid. But then we also think maybe that there's something in the cannabinoid pathways where some of those genes maybe have more affinity to different numbers of carbons in the side chains. So there's a lot more to learn. We have an application that's published for the genetic markers, and we're happy to discuss licensing. We're continuing to develop plants that have high levels of Varin. Just this last cycle, we got 2 more, or three more that have pretty high levels of Varin, 2 more 14% plants, and another 1-to-1 that's about 11%. And the kind of cool thing is that even though our Varin levels aren't going higher, the flower quality is getting better. So a lot of the Varin plants that have been out there have been tough to grow. They're sprawly, they make a lot of flower, but it's larvae, it's difficult to work with. And so we're getting to plants that look more like cannabis plants that you might want to produce from. And so it's pretty exciting. So I want to give full credit to the team that did all of this work. You see there with the long blonde hair and the black shirt, that's Erica Bacher. All of the computation, all of the statistics, that's all her work. She's incredible and has really brought to light all kinds of incredible findings. She's our senior director of gene discovery. The breeding team, the next 3 people there, John, Jared, and Becca. Jared's our director of breeding, and John's our VP. And they're directing a lot of the work so that we get the crosses done and making all those decisions using the markers. And then Amy on the far right leads the cultivation team. And without a solid cultivation team, none of this would happen. Grateful for them every day. It's been great hearing all about cultivation at this meeting and nutrients and just all kinds of the stuff that I want to take back to them. And then I also want to mention Sarah. She is our chemist and directs the lab and has been incredible to work with on all of these different cannabinoids and terpenes that we're studying. And so I want to give full credit. Without this team, none of it happens. I'm just the person up here talking. All right, I think that's it. So happy to take questions. Hey.
Hi, thank you very much for presenting. I noticed that on the recreational slide that you talked about THCV, that it mentioned the clear, energizing, focused effects. And one of the chemovars that's very well known for having THCV, Durban poison, in its terpene profile, its primary terpene is terpinolene, which also has the exact same description of effects. Was that including terpinolene, or was that THCV on its own, or how did that come about?
Yeah, so that's a great question. I didn't mention the dominant cannabinoids in the plants that we've been looking at. And actually, it's dominant in myrcene, which is not great.
It's terpinolene, limonene, and myrcene are the 3 dominant terpenes for Durban, I believe.
Yeah, so ours is myrcene and ocimene and beta-caryophyllene. So wow. You would not think uplifting, energizing at all. And so that's another focus of the program, is to put in those energizing terpenes.
Okay.
Yeah.
Thanks.
All right, thank you so much.